Temperature control is a recent development that provides an additional degree of freedom in studying nanochemistry by liquid cell transmission electron microscopy, notably the formation of gold nanoparticles in solution.
This methodology allows imaging of the dynamics of individual nanostructures in liquid with great control over the composition and temperature of the environment under realistic synthesis conditions.
Interestingly, these methods can be used to study the effects of temperature on the structural evolution of soft, hard, or biological nanoobjects in liquid environments by mimicking their formation or application medium.
The key success factors for liquid TM experiments are a clean sample preparation and a consideration for the electron beam effects on the nanoparticle dynamics.
For liquid cell preparation, first fill one glass petri dish with acetone and another with methanol in a fume hood. Place one small and one large E-chip into the acetone for 2 minutes, before moving both chips into the methanol for 2 minutes. After the methanol wash, use an air pistol and tweezers to dry the cells. And use a binocular magnifier or an optical microscope to verify the integrity of the silicon nitride window. If the chips are intact, plasma clean the E-chips with a mixture of argon and oxygen gas for 2 minutes. And load the gasket O-rings into the liquid cell holder. Place the small E-chip into the liquid cell holder and drop approximately 2 microliters of the liquid sample of interest onto the chip. Using a sharply cut piece of filter paper, remove any excess liquid from the chip until the liquid droplet forms a